Hello everyone. Today I will explain the concept of evolution of concept of organization of suit apex. The suit apex evolution concept of of suit apex. The suit apex the suit apical meristems are simple structure in vascular cryptogams. The apical cell is pyramidal in shape, with three or four faces pointing downwards and one face pointing upward, forming outer part of the plant. The apical cell divides in an orderly manner, has symmetrical division, and produces narrow flat cells. In seed plants, the shoot apical meristem does not show any distinctive cell. As in cryptograms, the apical meristem is supposed to be composed of two zones, outer tunica and inner corpus. They are distinguished from each other on the basis of plane of division. The cells of tunica region divide only with anticlinal walls perpendicular to the surface of tunica. The tunica layer grows as a sheet. When tunica is single layered, it is referred as monostratos, but when it is two or more layered, it is called multistratos. The corpus is covered over by tunica. The corpus consists of cells which divide in any direction, hence the region can grow in three dimensions. Apical cell theory. First one is apical cell theory. This theory was proposed by Nageli in 1878. It suggests the presence of single tetrahedral apical cell in the shoot apex in most of the vascular cryptograms. According to this theory, a single apical cell is the structural and functional unit of the apical meristem. This cell regulates the whole process of growth. This theory holds true for many algae, majority of bryophytes and pteridophytes. And second theory, histogen theory. This theory was proposed by Hanstein in 1868. Proposed by Hanstein in 1868. According to this theory, three meristematic zones have been recognized in the shoot apex of the angiosperms. Each layer possesses a unique set of initials, and these layers are referred as histogens. The outermost histogen is termed as dermatogen. जो outermost होता है, outermost dermatogen. The middle is referred to as peribleum. Middle peribleum. The innermost is referred as plerome. Each histogen performs a definite function. The dermatogen gives rise to epidermis, the peribleum gives rise to cortex, and the peribleum gives rise to cortex, will the plerome give rise to vascular cylinder. Later studies revealed that this histogen have no specific morphological function to perform. The theory failed because in most of gymnosperms and angiosperms, these three layers could not be distinguished. This was rejected later. And the third was tunica corpus theory. This was Tunica-Corpus theory, and the theory was proposed by Schmidt in 1924. Was proposed by Schmidt in 1924. The theory proposed that there are two distinct zones occurring in the shoot apex of the angiosperm. The peripheral layer consists of one or more layers of irregularly arranged cells named as tunica. The inner core of the cells surrounded by tunica form the corpus. The two divisions can be distinguished on the basis of plane of cell division. Tunica region comprises of cells characterized by anticlinal division. The cells of the corpus region divide in various planes. This theory does not provide any correlation between layers and formation of tissues. Tunica and corpus layer possesses separate initials. These cells are identified by their larger size. Vacuolated contents. The number of initials in two zones varies from species to species. The variations in number of tunica layers occurring in different species or even members of same species at different stages of development of shoot apex could result from plastochrome periodicity. Here noted 3 to 7 layers in the tunica region in Daphne pseudomeserum. Here is the name of scientist and noted 3 to 7 layers in the tunica region in 2 to 7 layers in the tunica region in Daphne pseudomeserum. These layers could be attributed to the seasonal growth changes. In some dicotyledonous families such as Malvasia, Laurasia, the outer layers of the corpus are uniform and resemble those of tunica. The stratified cells that sometimes show periclinal divisions have been considered part of the corpus. Thus, this concept become more flexible as the two regions which are treated as morphological entities are subject to fluctuations. Continuing, and second is continuing meristematic. Fourth, after that, continuing meristematic residue. The continuing meristematic residue is CMR. Continuing meristematic residue refers to the persistence group of meristematic cells in the shoot or root apex that remains undifferentiated over time, maintaining the capacity to generate new organs, leaves, branches, flowers. Will the surrounding cells proceed through differentiation, location, and composition? It is typically found in the central zone of the shoot apical meristem or root apical meristem. Shoot apical meristem or root apical meristem. Its location and composition. Significance of continuing meristematic residue. Maintenance of indeterminacy. Significance CMR. Continuing meristematic ratio. Maintenance of indeterminacy. Determinacy ensures continued organogenesis while preserving stem cell identity. In conclusion, continuing meristematic residue acts as the organizational hub of the apex, a slowly dividing center that sustains stem cell immortality, coordinates the performance of tunica and corpus layers, and ensures the indeterminate limit growth and orderly generation of new organs throughout the life of the plant. Its dynamics and structural organization differ among taxa, giving phylogenetic and functional significance. In essence, CMR is the engine for sustained organized growth, ensuring the shoot apex remains functional and capable of producing new plant parts throughout its life. After the last fifth theory is cytohistological zonation theory. Cytohistological zonation theory. The shoot apex of gymnosperms cannot be interpreted in the terms of tunica corpus theory. This is because it does not possess layer of cells that divide anticlinally. Foster interpreted the shoot apex of ginkgo biloba in terms of certain regions or zones which possesses distinct cytological characteristics. These zones have a relationship with the growth processes in the apex. The four growth zones have been recognized by him are given below. Like apical initial, group. This includes group of initials along the apical surface along with some lateral or sub-adjacent derivatives. And second is central mother cell zone. This zone lies below the apical initial group and is derived from it. The distal group of cells is lightly stained, vacuolated and shows slow activity. The primary walls of the central mother cells are thick-walled and pitted. And third is rib meristem. It consists of centrally located central mother cells and give rise to pith. The cells are vacuolated and divide transversely to form vertical files. This zone is also called as file or pith meristem. Also called file or pith meristem. Peripheral meristem. Flank meristem. This zone originates from the lateral derivatives of the apical initials and partly from the central mother cells. The cells are densely stained and mitotically active. The activity of this zone leads to the formation of leaf primordial. The pediclinal division in this zone causes elongation of the shoot, while anticlinal division increases the width. Now this complete. Thank you. Subscribe. Thank you. Like, share, and subscribe. Meet you in the next class, in which I will explain the root apex, evolution of root apex.